Dear students, in the last experiment, I told you how to make wet mount, yani aina bil moya. But in that experiment, we use letter E, not the actual cell, not the khali only letter E. Today, what we are doing in this experiment, today we will use the original cell. Cell we take from our mouth, filfam, cheek. Malum cheek? Check. Cheek is the inner part of the mouth. Okay, so we take a specimen from the cheek and we make the slide. Okay, 1 second. Okay, so we make the slide. Okay, so today we are going to study about human cheek cells and the differences between plant cell and animal cell. These are two objectives, two أهداف حق التجربة. Okay, in this picture you can see that one side we have a plant cell picture and the other side we have animal cell picture. So plant cell, animal cell. What is the difference between plant cell and animal cell? This slide is showing you the difference between plant cell and animal cell. Masalan, cell wall, Jadar al-Khalavi, cell wall present only in plant cell, not in animal cell. Plastids present only in plant cell, not in animal cell. But centriole and lysosomes, centrioles and lysosomes, they are present only in animal cells and not in plant cells. Okay, not in plant cells. Understand? Nucleus, Noah. Noah is present in both animal cells and plant cells, animal cells and plant cells, animal cell plant cell. But the difference في الحيوان Noah في المركز مركز الخلية. Noah is present in the center of the cell. On the other hand, in the plant cell, it is present at the corner. So these are some of the differences between plant cells and animal cells. You have to remember for the exam, because in the exam I can ask for the difference. What is the difference between plant cell and animal cell? Now, dear students, these are the ihtiyajat. These are the requirements for today's experiments. So today we are going to use toothpick, that you use in the mouth, snan, slides, cover slip, needle, qattara, yani dropper. But the most important thing to understand is this. Dear student, this is very important from the examination point of view, because in the exam I can ask this thing. What is this? This is the name of the sabagha. Today we are using a sabagha, yani stain. We want to give color to the cell, because our cells, they are shaffaf, transparent. We cannot see them in the microscope unless we give them some color. So this is the sabgha we use in this experiment. So in the exam, if I ask you what is the stain we use in the cheek cell experiment, so you will say methylene blue. Methylene blue is the name of the sabgha. You understand this thing? So now I am showing you the procedure. This is the procedure of today experiment. But this procedure, I am just showing you the video for this so that you can understand the procedure. Okay, so dear students, what we need for today experiment, cheek cell experiment. First thing is the slide, glass slide. Okay, second thing we use is toothpicks. Third thing we use is this water. And the last thing we use is sabagha, methylene blue. So just now I am starting. So he will take water. So he will put 1 or 2 drops of water. See, aina bil moya. one or two drops of water ala shariha. Now he will take toothpick, toothpick yani khashab asnan, and see now the procedure. The procedure is she will put it in her mouth, on the side of the mouth, inner side, and just roll. Do not prick, just roll. Okay, and then put this in the drop of water. Put this toothpick in the drop of water and mix. This will shift all the cells from the toothpick into the drop of water. And now throw the toothpick. Now this is methylene blue. I have told you the sabga. If you don't use this sabga, you cannot see the cells. So this methylene blue will give blue color to the cells, and then you can see the cells clearly. So she will just put two drops of methylene blue upon the slide. One or two drops of methylene blue upon the slide. Now the question of cover slip. Okay, I told you the method to put the cover slip. 45 درجة. بعدين شوية شوية نزل like this. Okay. Now in the last you will use mandil to absorb jazb, extra sabqa, sabqa zaid, from the corner, from the ends. Okay, just absorb if there is any extra sabqa, just to make the slide clean. Now now this is the complete slide. Okay, this is the complete slide. This slide we will put in the microscope. So starting from the low power, you will see the slide like this. These are all of these are cells. What cells? Cheek cells, your human cell. Okay, Kholiya insani. You can identify the cell by nucleus. Nucleus is present in the center of the cell. Okay, this is in low power. When you will increase the power, when you will increase the power of the microscope, you will see that they have become big. Okay, you can see many cells overlapping each other. You know, overlapping one over the other, you can see the cells. Okay, so there are many cells you can see in this picture. This is a big picture, zoom picture, and the last is this is the biggest picture of the cell. You can see that the margin of the cells, they are irregular, and there is a nucleus nova fil marcus in the center of the cells. And we do not find any cell wall, so this shows that this is a animal cell, and human cell is an example of animal cell. Okay, dear students, this is the procedure of the making of human cheek cells. Okay, same thing is shown in this picture. Yani, take the sample from the mouth, filfam, put it in water, mix, then add methylene blue and put the cover slip, and just see the specimen fil majhara. This slide is showing again the procedure I have told you already in the movie. Okay, so procedure. This slide is showing you the picture under low power and under high power. Okay, and I have told you that the cells are overlapping each other. The cells are overlapping each other. Remember, this is very important to understand. If the cells are overlapping each other, one over the other, how you will see them. This is we will discuss in our next experiment. Now, dear students,